goes up very quickly, huh? $21. We'll give another minute. $24. Okay. Good morning, everyone, and welcome to the Cancer Center Seminar Series. My name is Claudio Brunstein. I'm from the Division of Hematology, Oncology, and Transplant, and I'm here to introduce Dr. Armin Rashidi, who is a member of the Transplant and Cellular Therapy program at the Masonic Cancer Center. Dr. Rashidi has been a faculty at the University of Minnesota since 2016. Dr. Rashidi's initial training was in the medical school at the Tehran University of Medical Sciences, followed by a PhD in biogerontology and at the University of Newcastle University in England, followed by an internal medicine residency and a fellowship at WashU in hematology oncology. Dr. Rashidi has been a very active and productive member of our cancer center and our faculty. He's been involved in numerous clinical trials and translational research. More recently, he's dedicated his efforts to elucidate the mechanisms of in which the microbiome influences the outcomes of allogeneic transplant as well as the treatment of acute leukemia. He is very well published with 251 references and counting in PubMed. And he was recently awarded a KL2 award that is supporting his research in efforts to restore the microbiome with FMT, which is fecal matter transfer transplant. He's also the recipient of a Chainbreaker grant from our Masonic Cancer Center. Dr. Rashidi will speak to us today about the gut microbiota and infection complications of chemotherapy. Thank you, Rashidi.
Thank you very much. Good morning, everyone. I appreciate the opportunity. So yes, I'll be talking to you this morning about gut microbiota and infectious complications of chemotherapy in patients with leukemia mostly. So let's define dysbiosis for convenience and pretty loosely as disruptions in microbiota communities anywhere in the body. And our focus for today will be mostly on the gut. And remember, we have 10 or more times more microbial cells in our body than we have human cells. So we are really a super organism, a unity. We're not two different things separately from the microbes. And the questions we'll try to address to some extent today are: how much dysbiosis do we see in our cancer patients? How to characterize it? Does the dysbiosis resolve? And if it does, how quickly? Does it matter that we see dysbiosis in these patients? Can we prevent it? Can we fix it? And finally, what causes dysbiosis? I'll be talking about what we thought to be the main cause of this and what appears to be the cause based on the main cause based on some recent research. So the main line of research that I'm going to present today was really triggered, at least for me, by reading this paper, which is now almost 15, 16 years old, where the investigators asked the question, that during intensive chemotherapy for patients with acute leukemia, how can we prevent neutropenic fever? And it was known from a long time ago that neutropenic fever in an immunocompromised patient is dangerous because it's often a sign and the earliest sign of bloodstream infection, which could be rapidly fatal unless diagnosed and treated promptly. So the question and the hypothesis was, well, what if we give patients levofloxacin to prevent gram-negative bacteremias that at the time were a very, and still are a highly fatal complication of bloodstream infection in the immunocompromised host. So they randomized such patients to a levocoin group as well as a placebo group. And as you can see, there was a large improvement in outcomes, which was freedom from fever in the experimental group. So something like 20-25% more freedom from fever. But it's a very disappointing result, on the other hand, when you look at it carefully, because freedom from fever in the successful group is only about 35%. So 65% of these individuals are actually still developing fever, but yeah, that's an improvement compared to 90+ % who have fever. So this clearly suggested and indicated that we're missing something big here. Either those neutropenic fevers are not even infection. That's why levocoin isn't doing anything. The gap that we are not covering, the 65% gap, or they are refractory infections that are refractory to levocoin, and again, levocoin isn't doing anything, or there is just something else that's going on. And we know from practice that not all neutropenic fevers are bloodstream infection, and actually not all neutropenic fevers are even infection. So more than 70% of blood cultures in these patients who develop a neutropenic fever don't grow anything. It's quite interesting because we always assume the worst case scenario of a bloodstream infection, but we only find it in less than 25% of cases. And this clearly suggests that prophylaxis is highly empiric. Everyone is getting the same prophylactic antibiotic without us really expecting one or other infection, anything specific. And even worse, perhaps, is when they develop neutropenic fever, we don't know what causes it until 2-3 more days are passed. And by then, everyone has seen escalated antibiotics, and most of the dysbiosis we think has already been caused. And so if they come with an adverse clinical outcome, then it might be too late to do something about the damage that we cause to the microbiome. So because of this really high level of empiric prophylaxis and treatment that you don't often see in clinical settings, but we are completely empiric in this area. Some patients certainly benefit, as the NEJM paper showed, but some are likely harmed because we don't really know what we are targeting in those patients. And neutropenic fever is the central event. If we can prevent it better, so we don't need to give all patients the same antibiotic and then end up treating all of those patients with the same empiric antibiotic. Again, we probably will be able to prevent dysbiosis and its detrimental clinical outcomes that I will describe shortly. So understanding neutropenic fever pathogenesis better is key to this ideal personalized prophylaxis and treatment of this complication. So how did we do it here? We started 5 years ago with this very simple biorepository protocol in acute leukemia patients who were inpatient to get intensive chemotherapy. The first 20 patients collected stool samples and blood samples Monday, Wednesday, Friday on a calendar-driven basis, not infection or fever or any other clinical event-driven basis. And it was a little too intense for sample collection, and then we simplified it to just Monday and Thursdays for patients 21 and more. And we are up to 70 plus patients now. A huge biorepository of these samples have been collected, and they continued it until they were discharged from the hospital or 28 days after the beginning of chemotherapy. So since admission until about a month after starting chemotherapy. And these patients often are, well, they are always inpatient when they get intensive chemotherapy. They tend to get sick. A lot of them are sick. Almost everyone develops neutropenic fever. They all get a lot of antibiotics, heavy antibiotic pressure, prolonged hospitalization, 3 to 4 weeks, sometimes more of hospitalization, a lot of exposure to the nosocomial setting. Poor quality of life because of this and all other complications that we cause, expensive stay in the hospital. Everyone has a central venous access, which is a port of entry of bacteria. A lot of them develop mucosal damage in the mouth, in the gut, and in the areas where invasive procedure is done. And so that's another port of bacterial or other infectious agent entry. A lot of them have nutritional compromise, dietary changes because of nausea, vomiting, diarrhea, mucosal damage. They don't floss, and they are advised to use soft toothbrushes to keep the mucosal barrier of the mouth as intact as possible. And they all have private rooms. So this just summarizes the first 20 patients, and then the next 23 patients. Nothing really changed in the two, in the two cohorts in terms of standard clinical care for leukemia or hospital policies, with the exception of course, COVID precautions in the recent past, but pretty much everything stayed the same. So we felt comfortable combining these patients together. I say 23 because these are the most heavily analyzed patients now, but up to patient 70 plus, we've analyzed them. And some impressive findings here are that everyone developed a fever. So this is not following what the paper 15 years ago suggested. Freedom from fever is zero in these patients. And everyone is getting prophylaxis, including levaquin or other antibiotics. So again, clearly indicating we're missing something. And about a third of patients develop bloodstream infection, mostly gram-positive bacteria, as opposed to those days that were mostly gram-negative bacteria. And maybe that's why levaquin did a great job in preventing some of those fevers. C. diff infection, which is a hallmark complication of gut dysbiosis, happens in about a fifth or sixth of our patients, sometimes even higher. So this shows the really insane antibiotic pressure that these patients experience. So probability of exposure to each class of antibiotic over time aggregated among these patients is shown here. You can see the curve for fluoroquinolone is on top because almost everyone starts with that. And then it quickly drops while third or higher generation cephalosporins like cefepime curve goes up. And this is because these patients are spiking their first fever, so the fluoroquinolones are stopped, and then cefepime or other antibiotics are added on. So the curve stays, kind of at about a little bit of a plateau for fluoroquinolones for patients whose neutropenic fever happens later. But you can see with the rise in cefepime, with the rise in zosyn, with carbapenems, IV vancomycin, that almost everyone is developing some form of complication. And particularly if you look at oral vancomycin in gray, it has a steady rise close to the end of the hospitalization. The only indication for this drug in these patients is C. diff. So we are causing about a fifth of our patients are developing, or sixth of patients developing C. diff. And what happens at the end when the fluoroquinolone curve rises again? That's when patients have recovered their counts. A course of empiric or targeted antibiotics, such as cefepime, has been completed. So we're stopping those and putting patients back under levaquin prophylaxis until they are completely recovered. They go home, and then often we stop the antibiotic at that point, but we didn't continue that follow-up beyond 30 days. Now, how much dysbiosis we cause? The easiest way, but not the most accurate way to look at this is by looking at alpha diversity, which is just an intuitive way of thinking about this diversity. in a community of microbes, you get more of the different microbes and more at an even frequency. It's more diverse than a completely or almost completely dominated community, which is not diverse. And each point shows a sample here plotted with y-axis being a measure of diversity, x-axis being being day of sample collection relative to the first day of chemotherapy. So things start off in a few days before starting chemotherapy because we consented the patients early on and started collecting these samples. And then points that are connected to each other belong to the same, or samples belonging to the same patients. So a healthy person has about, a diversity of 4 or so. But these patients start there or almost there and then quickly drop their diversity. So acute leukemia patients are red compared to allogeneic stem cell transplant patients who develop even more severe dysbiosis. These are pretty extreme. You can see that some patients with leukemia, their diversity drops to 0. That means there's just one bacteria in the gut, and it actually happens to be a lot of the times Enterococcus. And it's not just diversity, but it's also the composition of the microbiome that departs from the baseline composition a lot during this chemotherapy. So patients who are experiencing the first antibiotic and chemotherapy and hospitalization insult are shown in red, and those who've already seen this and the leukemia has relapsed and they're undergoing a second round of chemotherapy are shown in blue. And what we are showing here is similarity of the microbiome composition as a community to its own baseline state. So everyone in the beginning is 100% similar to their own baseline, of course. But this drops very quickly, and those who've already had some exposure and some experience in the past develop even more severe departures from their baseline, suggesting that the ecosystem of the gut and the host remembers prior injury, and it behaves differently if it's already been injured, that it's naive to such insults. So there's a lot of damage. It's most prominent when you look at diversity, but also there is a significant compositional change. How to characterize it? There is something interesting that happens in these patients. So if you look at this heat map of the 20 most abundant taxa across all samples, and with samples shown as columns and general abundances shown as rows, with light yellow being lower abundance, and as you get more and more red and dark, you're looking at higher abundance. And with these and with this sidebar showing sample collection day with light colors being earlier days, so right at the time of admission, for example, is white. And then as you move towards day 30, these bands become darker. You can see that some of these most common bacteria actually belong to the mouth. So Rotia, for example, a lot of the streptococci, or for example, Veillonella. These are most commonly, and some Veillonella are bacteria that you expect them in the mouth, but you're seeing them in the gut. And so it seems the colonization resistance in the gut is impaired because of antibiotics or everything else that's happening to these patients. And then when they swallow their saliva, those that couldn't in the past colonize successfully in the gut are now managing to stay and survive there. And who knows what they do to the host? It's completely unexplored territory. So clearly something very wrong and abnormal is happening in these patients. And then these communities, gut communities, start to get dominated by some pathogens. So looking at induction therapy versus repeat therapy patients, reinduction chemotherapy patients, for example, going from their baseline sample towards week 4, you can see there is an expansion of the orange bar, which is Enterococcus, and it's so much more severe in patients who've been damaged before. So that's scar idea. So almost half of, or even more than half of the microbiome is occupied by Enterococcus. This shows the quantitation of the same picture where error bars that are completely under the regression coefficient 0 show that that particular bacteria is associated, so the abundance of that bacteria is inversely associated with diversity. Therefore, in multivariable regression, the coefficient of regression for that variable is negative, and the 99% confidence interval for that is also completely below 0. And that's the only bacteria that it's dominant in low diversity communities, with a close competitor being lactobacillus. So Enterococcus is the dominant bacteria or genus in low diversity samples in these patients. Does this dysbiosis resolve, and how quickly? I won't spend much time here today, but these are 3 patients, each column being a patient, who actually contributed samples to a concurrent and subsequent biorepository in transplant patients, because these 3 patients participated in the leukemia biorepository, went into a remission, and then moved out to transplant, and then contributed samples on that biorepository. And so we have this really unique and really beautiful biorepository, longitudinal, spanning over many months. And so you can now see how the dysbiosis diversity composition change from baseline evolves over time with each round of antibiotic and other types of damage. And instead of focusing on this, what I'm just going to tell you is that while diversity fluctuates and often resolves to or returns to its baseline good level, say 3.5 or so, after we give patients some time. Although diversity does this, composition often does it. So it looks like a diverse community again, but it's not the same community. It's often a very pathogenic community full of pathogens, which looks just equally diverse, but it's not a good community anymore. And this takes us to the to the concept of stability, resilience, and resistance. So imagine this is your fitness landscape for a community, and you are here in a healthy state. You perturb it a little bit. For example, you stop eating meat or stop eating vegetables, or you take a brief course of antibiotics, you fluctuate it a little bit, it kind of goes into the spiral and ends up in the same way. Or you start to perturb it really badly with more antibiotics or some more pressure or dietary change, moves up this landscape, but you stop that perturbation, it goes back down to its healthy state. But there is a limit. If you keep pushing it away from its healthy state, it can reach this peak where it's no longer stable, and it only takes its tiny touch, and it's the tipping point. It falls down the other side of this mountain into another steady state, which is no longer the same. It's far away from that, and it could be a very pathogenic, unhealthy state. We don't know what these things mean, really. We don't know what's a healthy state, what's an unhealthy state, except we assume when you have a lot of pathogens with antibiotic-resistant genes and patients start to have clinical complications, it's presumably an unhealthy state. So those 3 patients that I showed you show both of these or all of these phenomena. Resilience, how much perturbation it takes to completely deviate the ecosystem to a new steady state, which could be very, very different. These are really theoretical and ecological concepts that are really fertile ground for investigation in the microbiome field. But does it really matter? Does it matter if we see dysbiosis in these patients? We know C. diff, we've known it for a long time. That's a hallmark complication of dysbiosis. Also pathogen colonization with antibiotic-resistant organisms such as vancomycin-resistant enterococci. Lower respiratory tract infection, the gut-lung axis, so-called axis, bloodstream infection, mucositis, time to hematopoietic recovery, all of these have been associated with disruptions in microbial communities in the gut. And this is the conventional model that was actually worked on heavily here in the University of Minnesota many years ago, where where chemotherapy, and it's still true that chemotherapy immunocompromises the host. It also damages the gut barrier that separates the blood and the host from the gut luminal microbes. And by damaging this, there is a port of entry now that's opened up for these bacteria or other pathogens to translocate to the bloodstream. But it seems the conventional model needs some revision here. And the revision involves the gut microbiome and its dynamics. So with antibiotics, nutritional changes, perhaps even chemotherapy, we are disrupting these communities in the gut, which could then influence the gut barrier damage that chemotherapy causes and contribute to sepsis, infection, and other complications of therapy. In addition, dysbiosis itself may actually trigger some pyrogenic pathways within the host without causing any infection. And that's when you think, oh, so maybe some of these neutropenic fevers that we've kept assuming are infection are not in fact any infection. Maybe they're just triggering, cytokine cascades that stimulate the thermoregulatory centers and cause fever without any infection. So we thought, well, let's have a biomarker for neutropenic fever that we can quantify it and then correlate it with gut microbiome. And we chose flagellin because it's easily measurable by ELISA in the serum, and it comes from flagella, which is the locomotor machinery of motile bacteria. Yes, of course, it doesn't reflect non-motile bacteria such as Enterococcus or Streptococcus, but it captures a lot of bacteria and bacterial translocation events in the blood. So if you look at this, the flagellin, and this is log transform, so it really peaks in the flagellin, the serum peaks in the midst of chemotherapy in the middle of the hospitalization, when all of these red dots show neutropenic fever episodes, and then it gradually comes down. So an okay biomarker of neutropenic fever, and it's much higher, again, log transformed, much higher in around the days of neutropenic fever than before neutropenic, than days without neutropenic fever. So if you then correlate, it's more complicated than just a correlation, but in regression, you try to correlate the levels of flagellin with different genera in the gut, as well as clinical factors. You find that in the 7 days and 5 days preceding each neutropenic fever, there is an expansion of lactobacillus, but more importantly, Akkermansia in the gut. So Akkermansia is a mucolytic genus in the gut. It feeds on mucin. So by thinning the mucus, it can actually weaken the gut barrier, and that promotes translocation of other bacteria. Akkermansia by itself doesn't translocate as far as we know, but it just opens the ports for other bacteria to get in. Therefore, the alternative model that's also visualized here. So the current paradigm was or is that whatever expands in the gut will go in. The new model says some of what expands could actually just weaken the gut barrier, and then other bacteria that haven't even expanded have a higher chance to get in, particular bacteria that are more virulent for passing through a weakened gut barrier, and then they can cause sepsis and complications. We validated this in the rest of our patients. Everything I showed was about the first 20 patients. And if you do this bioinformatic analysis called LEFSI and look at gut stool samples that were collected shortly before a neutropenic fever, and you call those samples high risk versus other samples that are low risk, you can see Akkermansia is the top genus that characterizes high risk samples. And you can again quantify this. This is not just 7 days, it could be 5 days, it could even be 3 days. And Akkermansia is higher in samples that precede neutropenic fever. And it doesn't matter if you have low diversity or high diversity in the rest of your microbiome, the relationship between Akkermansia on the x-axis and the probability of neutropenic fever on the y-axis is identical. So possibly showing a direct genus level or organism level association with a clinical complication. And this paper was published a few years ago in a mouse model, but you can easily extrapolate it to humans, I think, that when the individual is having a fiber-rich diet, the majority of the bacteria will use those dietary carbohydrates as the main source of their nutrition. And those bacteria are shown in green, fiber-degrading microbiome, and there are some pathogens shown in yellow, and there is Akkermansia or mucus-degrading microbiome shown in red, the spiked bacteria. So if you deprive patients or individuals from fiber, dietary fiber, then the bacteria that succeed in this survival competition are those that have an alternative source of nutrition, i.e. mucin, shown in pink here. So the Akkermansia, for example, those bacteria will expand at the cost of fiber-degrading bacteria that have to die. And then when Akkermansia expands, it eats them, it erodes into the mucin, and then the pathogens have an easier way accessing the gut barrier. And imagine the gut barrier being also damaged by chemotherapy, and then they can get into the blood. So neutropenic diet is a diet that's depleted of fiber, fresh fruits, vegetables. Probably not a good idea in these patients, although one needs a randomized trial to show it. And parenteral nutrition, again, not letting patients eat particularly fiber, may not be the best idea in these patients. Now, we wanted to see what actually mediates the pathway from starting with Akkermansia and ending up in neutropenic fever. An obvious hypothesis was that, well, maybe there are some metabolites that diffuse from the lumen, metabolites either from Akkermansia or from other bacteria that diffuse through this damaged gut barrier to the blood, and those metabolites may trigger inflammatory and pyrogenic pathways. So then first we thought, well, can we even distinguish neutropenic fever samples? That means serum samples that were collected within 24 hours before a new episode of neutropenic. Can we distinguish those from serum samples that didn't precede a new episode of neutropenic fever, just using serum metabolites? And by this metabolomic analysis, we find that you can indeed distinguish these two groups of samples, those with neutropenic fever shown in red and non-neutropenic fever in blue, by a select set of these metabolites with a very good accuracy. And those metabolites are shown here, blue being metabolites that are associated with are not associated with neutropenic fever, and red being those that are associated with neutropenic fever. So this would just show that, yes, the metabolomic profile of blood or serum differs between samples with versus without neutropenic fever. And then we thought, can we find metabolites in the serum that are associated with Akkermansia expansion in the gut? Because then if a subset overlaps between these two metabolites, then you can say these metabolites are probably mediating the association between Akkermansia expansion and neutropenic fever. So this volcano plot shows the association between Akkermansia in a stool sample and metabolites in the serum within the next 7 days. Because it might take some time for microbial changes in the gut to cause a change in serum metabolites. So we just assumed 7 days, and then as sensitivity analysis, we also looked at 5 days and 3 days. And what this shows is what metabolites in blood are significantly associated with Akkermansia in the gut in the preceding several days. So each point is a metabolite, and colors indicates the pathway that that metabolite is involved in. If a circle or a dot is above this line, it's highly significant. And if it's to the right of this vertical line, it's positively associated with Akkermansia. If on the left, it's negative. So our interest are these metabolites. Those on the right are positive associated with Akkermansia, on the left, negative. And then you zoom on these metabolites only and see which ones, so on the right, which ones of these selected metabolites are actually associated with neutropenic fever in the next 24 hours. And again, those above this line, you are limiting it to just about 8 metabolites that are strong and significant associates of both neutropenic fever and Akkermansia. But now you need to be careful, because you just need to select metabolites that are either positively associated with both phenomena or negatively with both. Otherwise, a positive one side, negative the other side wouldn't be an indication of mediation of an association. It has to be the same direction. And that brings you down to 3 metabolites: cysteine glycine, DHEAS, and beta-cetyl glutamate. Now, I'm going to focus on the first one, just on the cysteine glycine. What is this? So this is a cell, and GSH is glutathione, the main intracellular antioxidant molecule that we have in our body. So when there is oxidative stress, glutathione counters it and prevents oxidative damage. So the glutathione can be exported to the outside of the cell, where it has a very short half-life, because this gamma-glutamyl transferase, GGT, on the surface of the cell quickly degrades glutathione to a glutamyl moiety, which then is bound to various amino acids and cysteine glycine, our molecule of interest. And then a dipeptidase on the surface of the cell breaks cysteine glycine down to glycine and cysteine. Cysteine and glycine are then taken up by the cell, and the glutamyl amino acids go back to the cell. Glutamate is released from it, and glutamine, cysteine, and glycine are merged, combined to replenish the glutathione source in the cell. So now, if the glutathione is exhausted because there is just so much oxidative stress, then you can imagine the body upregulates the GGT so that any glutathione that happens to be in the extracellular space is immediately degraded to cysteine glycine and this glutamyl amino acids. So then the constituents of the cysteine glycine are produced and immediately taken back to the cell to replenish glutathione. Now, what we found was cysteine glycine is high in association with both neutropenic fever and Akkermansia, but glycine and cysteine are both extremely low in those high-risk samples, and glutamyl amino acid is also low. So what does this whole thing mean? It means, most likely, this means the body is trying to make a lot of cysteine glycine, but whatever is generated from cysteine glycine is quickly used by the cell to as a defense mechanism. And the same for glutamyl acid. It's a component that's eventually going to be used for glutathione resynthesis. So overall, suggestive of a oxidative stress state in the body before neutropenic fever happens, suggesting that perhaps Akkermansia expansion increases oxidative stress in the blood, in circulation. And we know that the CNS and thermoregulatory center are sensitive to oxidative stress, and actually pyrogenic pathways, prostaglandins, can be stimulated by increased oxidative stress. Now, this was a very curious question. Why not just sequence the entire, look at the entire sequencing of the microbiome in the serum, and instead of making guesses, just find out if during neutropenic fever, before or after, there is a different bacterial composition in the blood. This is, of course, sequences. We don't know if DNA we find means live bacteria, but they were live bacteria at some point, presumably. And this just shows the distribution of these bacterial sequences in the blood by 16S amplicon sequencing. But interestingly, with time, again, same definition of sample collection day, the diversity of the serum microbiome doesn't really change. And more strikingly, the composition of the serum microbiome doesn't change whether you are before fever, after fever, or on the day of fever if your serum was collected. This shows a principal component analysis that samples really don't segregate based on microbiome composition. So that kind of suggests that maybe a lot of what we think is infection, in the blood at least, it's not infection. Now, how can we prevent it? Preventing dysbiosis, one could just be more cautious with antibiotic use. De-escalate them when you don't need them. Discontinue them when you don't need them. But there is no consensus. It's just such a hard clinical decision to make when we don't know what causes fever to assume, well, it's not infection, let's go down on antibiotics. It's difficult to make this decision clinically. So that really hasn't gone too far in clinical care. But one could use these experimental agents at this point that are oral drugs that degrade antibiotics that reach the lumen of the colon. So these are non-absorbable. But when, for example, a cephalosporin that's given IV for a purpose, for say cholecystitis, for example, when the fraction of it is reaches the lumen of the colon, then these non-absorbable beta-lactamases will degrade it and don't let them damage the microbiome. And there are various compounds that are in different clinical trials, and some have moved forward to even phase 2 trials. What we've done here is a very simple thing: activated charcoal, oral powder that we want to give eventually to patients who are getting IV antibiotics, because charcoal used for poisoning, as you know, is such a strong absorbent that when it reaches the colon, and it's non-absorbable, by the way, when it reaches the colon, it stays there and binds to antibiotics that are in the lumen and saves the microbiome. So we found an optimal dose and solution in healthy volunteers in a trial here, and we are moving shortly to testing it in lung cancer patients who are getting mediastinal staging, and before that, they are getting a single dose of IV antibiotics. We want them to drink charcoal to protect their gut microbiome. And of course, one can manipulate the diet. One can try neutropenic versus regular diet. Again, very little consensus on how to do these major components of clinical care. How can we fix the dysbiosis? So one could use prebiotics, for example, dietary fiber, things that help beneficial bacteria grow. And potato starch is the top candidate based on a successful, recently published clinical trial. One could use probiotics, giving select bacteria to the host, hoping that it will survive in the community, in a new community, and somehow adjust or fix that community, or at least survive and grow there and offer the host some benefits. Limited success in at least our patients. Or one could try to replace the entire sick microbial community by transferring a healthy, presumably healthy microbial community from another donor. A healthy person would donate a stool sample, and we could give those microbes to a patient with a damaged microbiome. Obviously, this is transferring microbes, so safety in an immunocompromised patient is a concern. And this paper was published from a side effect that drug-resistant E. coli bacteremia was transmitted by fecal microbiota transplant. It could be traced at the level of the strain to the product, to a patient who was heavily immunosuppressed, and to 1 or 2 patients, and they died with ESBL E. coli bacteremia. One could collect it from the patient, perhaps before they were sick, before they were exposed to antibiotics. Very hard because we don't know beforehand who's going to be a patient when they are healthy. And if you collect it late, then you're transferring a sick microbiome to a different sick microbiome. It may not work. Or you could collect it allogeneically, as I just mentioned. You could give it as through a nasogastric or nasoduodenal tube, and that's how it was done in the past. Shown on the left, it's a bag of stool. Or you could do it in a smarter way, as with enema, or in oral as oral capsules, like we do in our trials here, shown on the right. This is a trial that was successful in prebiotics in transplant, stem cell transplant population, and it had a nice immunomodulatory effect in a beneficial way, reducing graft versus host disease, and also mitigated some of the mucosal injury that radiation and chemotherapy preparative regimen caused in these patients. The investigators showed that as a result of growth of beneficial bacteria, butyrogenic bacteria in the gut, in the experimental group who got prebiotics, this was potato starch, the butyrate level in the gut as well as diversity of microbiome increased. These are the largest FMT fecal microbiome transplant studies in the bone marrow transplant patients or acute leukemia patients. Sample sizes varied between 10 and 25. Most of them had a microbiome primary endpoint because it was hard to imagine and actually do a trial with a clinical endpoint. You would need a large sample size, but we've done it. We've started to do it here. So in this trial that is ongoing here at the U, we have 72. We are enrolling 72 AML patients and 72 allogeneic stem cell transplant patients in a phase 2 randomized double-blind placebo-controlled trial where we give allogeneic encapsulated FMT from healthy donors. And Alex Koretz has been preparing these capsules and providing us with those capsules. And we give these capsules repeatedly. So after each antibiotic exposure, after hematopoietic and immune recovery in these patients, we give a dose of these because we know the damage has been caused, but antibiotics are gone now. So we can safely give the capsules without them being destroyed by ongoing antibiotics. And we do it up to 3 times after 3 months, for the first 3 months after enrollment. And our clinical endpoint is infection, because we have enough preliminary data to show that the gut microbiome actually modulates infectious complications. So instead of having microbiome endpoints as primary endpoint, we have all-cause infection as the primary endpoint. And of course, our stopping rule is a major safety concern that other trials have shown, and that's bloodstream infection within 7 days of giving FMT. We haven't seen it in our patients, as I will show in the slide. So far, we've had 32 patients. The trial is going well. The only possibly related severe adverse events, those have been rare, but we've seen 1 case of gut acute GVHD grade 3. I think it's unlikely to be related, but we don't know for sure. There was 1 case of E. coli bloodstream infection grade 3. This is blinded, so I don't know if the patient actually got placebo or FMT. This has been an only event. CMV viremia in 2 cases within 7 days of the product, and our E. coli bacteremia was not within 7 days of the product. So what causes dysbiosis? I'll just try to spend a couple of minutes on this. We looked at the host genotype in transplant patients because they are more or less similar to our leukemia patients. But what I'm going to show you here is actually data from healthy adults, from 250 healthy adults in the Human Microbiome Project. And we show that a single nucleotide polymorphism in the alpha defensin 5, this is a host SNP, is associated with altered levels of Odoribacterus splanchnicus, an obligate anaerobe in the gut of these healthy individuals. And now, what's alpha defensin 5? It's the most abundant antimicrobial peptide that's secreted by these Paneth cells in deepest part of intestinal crypts. So the function of these antimicrobial peptides is actually to kill the microbes and modulate the gut microbiome in the vicinity within the lumen. So it's then quite possible that the host genotype is playing a role in determining the gut microbiome. So that's potentially one cause. We thought, well, almost all patients are getting levoquin. Maybe levoquin is the cause of these microbiome alterations. What proved 15 years ago to be a beneficial thing, maybe it's not beneficial. So we compared samples that had no antibacterial antibiotic exposure before patients were exposed to their first antibiotic, levoquin, compared them to those samples between levoquin and escalated antibiotics when patients develop neutropenic fever. So in this period, the only antibacterial exposure was levoquin. So essentially, by comparing these two samples, we are looking at the effect of levoquin, plus, of course, diet and other factors. So in multivariable analysis, it turns out that levoquin wasn't that detrimental. So Blaudia was one of the two species, genera that was altered with levoquin, but it increased, and it's a beneficial microbes. The only one that decreased, and we don't know what it really does in our patients, is Parabacteroides, but by and large, I think, it didn't do much harm to the gut microbiome, so that's probably not the cause of dysbiosis. But how about the baseline microbiota? Could the baseline microbiota impact how the microbiome responds to such antibiotic pressure? And we don't know that. No one's done this type of study before. With single dose, with a brief course of a couple of antibiotics, the pretreatment microbiome has been established as a determinant of microbiome trajectory. But what if we give all of these antibiotics? So first, we looked at our samples and just looked at the dissimilarity, the difference between composition of microbiome in every two samples collected from the same patient. So every circle in this shows one such comparison, the difference in microbiome composition between those two samples. And as those samples are more separated along time, you can see that the difference between two is going up. Kind of obvious and expected, because with time, there is more and more of different insults, antibiotics, nutrition, chemotherapy. So you expect the microbiome to change, of course. So this was a proof of concept here. But then when you look at how the baseline microbiome, shown in red, the antibiotic exposure history of each sample, shown in red, contributes to subsequent to the sample microbiome, and then you compare it to how the baseline microbiome from that patient contributes to the microbiome in subsequent samples, and how both of these factors together predict the microbiome in subsequent samples. You can see that, interestingly, the antibiotic history of each sample is not a major determinant of that sample's microbiome. Quite striking. In contrast, the baseline microbiome for that patient is a much more significant predictor of subsequent samples from that patient. And of course, when you combine them both, you get a stronger model, and you get up to predictions of something like even 60–62% accuracy in predicting the top 20 genera across all samples. This is very interesting, because now not only you can see what's more important in determining microbiome composition, but you can also tell which genera are most predictable. So for example, here, genus 1 is Bacteroides, is very strong. Well, it's moderately strongly predicted just by the baseline microbiome, but pretty poorly by the antibiotic history. Enterococcus, shown in 12, is not well predicted by the baseline microbiome. Rather, it's more strongly predicted by what antibiotics we are giving the patient. So a lot of information from this rich graph that you can obtain. I will skip this for the sake of time, but what this is showing is that the baseline microbiome, there are certain genera within it that make the largest contribution to subsequent samples, subsequent microbiota trajectory, and those happen to be the obligate anaerobes of the gut that make short-chain fatty acids such as butyrate. So in conclusion, microbiota may be a neglected key to precision therapeutics. It can be a predictive biomarker of response to intensive therapy and antibiotics. Empiric prophylaxis and treatment using antibiotics is in need for a revision. This is a major problem. And I think sometime in future, this is what we need to have. You get a baseline sample from a patient before you start damaging their microbiome with various factors. That can help you determine how to treat that patient, how to give them chemotherapy, how to give them antibiotics. You can try to prevent damage to that microbiome. You can even try to optimize that microbiome if you have time with prebiotics, probiotics, FMT. And then while you are giving the patient treatment, you can modify it based on how the microbiome changes or what information you learned from the baseline microbiome. You can prevent further damage. And even at the end, once you're done with your treatment, say leukemia treatment or transplant, the information you get from microbiome can help you determine the follow-up and try to fix the damage that you inevitably caused during this process. Therefore, you will have your desired outcome and desired microbiome. These are my acknowledgements, and thank you very much.
Okay, we have a question from Sabri, so I'm going to allow you to talk, Sabri. Your hand is up. Go ahead.
Can you hear me?
Yes.
Yeah, Armin, like a wonderful presentation, lot of background information. This is really helpful to understand the dynamics of gut microbiome and then what possibilities we have. So one understanding is this complex bacteria, it's not just one bacteria, it's a community of bacteria. And a part of the community can be like kind of disrupted, that leading to things where changes in the metabolic composition, and that metabolic composition can indirectly or directly affect a lot of downstream events. So one side of the equation is the microbiome itself and the part of the community. The other side is the metabolites coming from the microbes. Can we think of identifying the composition of these metabolites and for replacement therapies or like metabolite-based therapies, or a hybrid of both combination of microbiome metabolite therapies?
Yes.
Well, what's your thought on this?
Yeah, it's a very good question. There are actually bioinformatic models recently developed to really combine microbiome with metabolome, even with transcriptome, with proteome, all of these omics, high throughput data, just combine all of them together. And then you can tell, okay, this bacteria is associated with this RNA, with this protein, with this metabolite, and with this clinical outcome. And then the question is, okay, how can we fix one of these without changing the entire community in a detrimental way? So for example, with parenteral nutrition, we can add metabolites. We can add a bunch of metabolites to it, but how would it impact that community of metabolites? This is a very difficult question, and I think there's just such a long way to go to address this. I think computational modeling, just predictive modeling, and doing germ-free mouse experiments, very controlled environments when you can really manipulate individual components of the system is important. Translating it eventually to patient care and humans is, I don't think it's going to be easy. So.
Yeah, that's helpful. So it's all starting points, right?
Yes. Yes.
Great. Thank you.
And we're right at 10 o'clock, so I don't see any other questions. So I think that's it for today. Thank you, Dr. Rashidi,
Thank
for your talk. It's been great. Thanks, everybody. See you next week.